It will result in allergens and toxins in our food. Well, again, the great advantage of today's transgenic technologies is the precision they allow us in determining how we change the plant. Aware that certain substances tend to provoke allergic reactions, we can accordingly avoid them. But this concern persists, stemming to some degree from an oft-told tale about the addition of a Brazil nut protein to soybeans. It was a well-intentioned undertaking. The West African diet is often deficient in methionine, an amino acid abundant in a protein produced by Brazil nuts. It seemed a sensible solution to insert the gene for the protein into West Africa's soybean. But then someone remembered that there is a common allergic reaction to Brazil nut proteins that can have serious consequences, so the project was shelved. Obviously, the scientists involved had no intention of unleashing a new food that would promptly send thousands of people into anaphylactic shock. They halted the project once the serious drawbacks were appreciated. But for most commentators, it was an instance of molecular engineers playing with fire, heedless of the consequences. In principle, genetic engineering can actually reduce the instance of allergens in food. Perhaps the Brazil nut itself will one day be available free of the protein that was deemed unsafe to import into the soybean. It is indiscriminate and will result in harm to non-target species. In 1999, a now famous study showed that monarch butterfly caterpillars, feeding on leaves heavily dusted with pollen from Bt corn, were prone to perish. Well, this was scarcely surprising. Bt pollen contains the Bt gene and therefore the Bt toxin, and the toxin is intentionally lethal to insects. But everyone loves butterflies, and so environmentalists opposed to GM foods had found an icon. Would the monarch, they wondered, be but the first of many inadvertent victims of GM technology? Upon examination, the experimental conditions under which the caterpillars were tested were found to be so extreme, the levels of the Bt pollen so high, as to tell us virtually nothing of practical value about the likely mortality of caterpillar populations in nature. Indeed, further study has suggested that the impact of Bt plants on the monarch and other non-target insects is trivial. But even if it were not, we should ask how it might compare with the effects of the traditional non-GM alternative, pesticides. As we have seen in the absence of GM methods, these substances must be applied liberally if we are to have agriculture that is as productive as modern society requires. Whereas the toxin built into Bt plants affects only those insects that actually feed off the plant tissue, and to some lesser degree, insects exposed to Bt pollen, pesticides unambiguously affect all insects exposed, pest and non-pest alike. The monarch butterfly, were it capable of weighing in on the debate, would assuredly cast its vote in favor of Bt corn. It will lead to an environmental meltdown with the rise of superweeds. The worry here is that genes for herbicide resistance, like those in Roundup Ready plants, will migrate out of the crop genome into that of the weed population through interspecies hybridization. This is not inconceivable, but it is unlikely to occur on a wide scale for the following reason. Interspecies hybrids tend to be feeble creatures, not well equipped for survival. This is especially true when one of the species is a domesticated variety bred to thrive only when mollycoddled by a farmer. Let us suppose, for argument's sake, that the resistance gene does enter the weed population and is sustained there. It would not actually be the end of the world or even of agriculture, but rather an instance of something that has occurred frequently in the history of farming. Resistance arising in pest species in response to attempts to eradicate them. The most famous example is the evolution of resistance to DDT in pest insects. In applying a pesticide, a farmer is exerting strong natural selection in favor of resistance, and evolution, we know, is a subtle and able foe. Resistance arises readily. The result is that the scientists have to go back to the drawing board and come up with a new pesticide or herbicide, one to which the target species is not resistant. The whole evolutionary cycle will then run its course before culminating once more in the evolution of resistance in the target species. The acquisition of resistance, therefore, is the potential undoing of virtually all attempts to control pests. It is by no means peculiar to GM strategies. It's simply the bell that signals the next round and summons human ingenuity to invent anew. Despite her concern about the impact of multinational corporations on farmers in countries like India, Suman Sahai of the New Delhi-based Gene Campaign has pointed out that the GM food controversy is a feature of societies for which food is not a life-and-death issue. In India, where people literally starve to death, as Sahai points out, up to 60% of fruit grown in hill regions rots before it reaches market. Just imagine the potential good of a technology that delays ripening, like the one used to create the Flavor Saver tomato. The most important role of GM foods may lie in the salvation they offer developing regions, where surging birth rates and the pressure to produce on the limited available arable land lead to an overuse of pesticides and herbicides, with devastating effects upon both the environment and the farmers applying them, where nutritional deficiencies are a way of life, and too often of death, and where the destruction of one crop by a pest can be a literal death sentence